Okay, so DNA stands for deoxyribonucleic acid, and your DNA structure is made up of what we call DNA nucleotides. So a nucleotide is made up of a phosphate, a sugar, and a base. So your phosphate group links to the sugar, which is deoxyribose, okay, because it's missing an oxygen there. And your base can either be adenine, cytosine, guanine, and thymine. So we call them the nitrogenous bases, and they will look something like that. Okay, your adenine is double ringed, okay, with your nitrogen in there. Similar to guanine, except as you can see there, in adenine we've got an N, whereas there we've got an O in our bases, as well as extra N over there. So they're similar, but they are different. But they have two rings. And then your cytosine and your thymine, they have just one ring. Similar structure, but again, different in terms of where the nitrogens and oxygens are. So the double ring structures we call purines, and the single ring structures we call pyrimidines. And a purine will always pair with a pyrimidine, okay, because we need our DNA structure to be even, so we always have a double ring paired with a single ring. So DNA is going to be double-stranded, and you're going to have all the nucleotides linked together, where we have our one nucleotide joined to the next one, and then on the other side we have the same thing with the bases meeting in the middle. So now, as I said, a purine is always going to match with the pyrimidine so that this is nice and even. So we're going to have our adenine will always pair with thymine, and guanine will always pair with cytosine. So we always have A and T and G and C. Now those letters could be in any order on any side, but as soon as there's an A on one side, it means a T will be on the other, or if there's a T on this side, A will be there, and the same for G and C. So now these are linked in the middle by hydrogen bonds, and there is 2 hydrogen bonds between adenine and thymine, and there is 3 hydrogen bonds between guanine and cytosine. So we have hydrogen bonds holding in the middle, and then to connect your sugar phosphate backbone for your one nucleotide to the other, that bond is called a phosphodiester bond. So that would then be our structure of DNA with your phosphate, your sugar, and your base all linked to each other by a phosphodiester bond to create what we call over here a sugar phosphate backbone, held together in the middle by the bases, which we call complementary base paired, because A is always with T and G is always with C. So those are complementary base pairings held together by hydrogen bonds in the middle. So before we can undergo cell division, what we need to do is replicate our DNA, because we need to have 2 exact copies of the DNA so that each can go to one cell. So if we look at the process of DNA replication. So DNA replication, I'm going to take a strand of DNA that I have with my sugar phosphate backbone, and then I have my bases in the middle held together by hydrogen bonds. So what I'm going to do is I'm going to use each strand as a template. And what happens is the DNA splits and we break the hydrogen bonds in the middle. And while it's being split, down the middle, it's going to keep going. At the same time, we are going to get new nucleotides that will then join to each side in there. So we're going to have, at the same time, both nucleotides on each side are going to come in and they are going to start to join each side, all while we are still splitting. So you end up with these fragments that get joined together by the hydrogen bonds with enzymes, and then an enzyme is going to join the backbones together so that we end up with this entire single strand on that side and a new strand on this side. So we keep going the whole way as it keeps splitting, so that in the end you end up with two completely new strands. So if you look at that in a diagram, because the DNA is so long, this happens in whole different sections until eventually it all meets up and there's one whole new strand. So where we keep adding, we call that at the replication fork. And there's multiple replication forks all the way through a strand. Okay, and what happens at that replication fork is we have an enzyme that is breaking the hydrogen bonds, and that enzyme is called helicase. So helicase breaks the hydrogen bonds, as I just showed you. We have our new nucleotides coming in fragments, and we call those the Okazaki fragments. Okay, so the Okazaki fragments come in and they complementary base pair and match to the one strand that we call the template. And DNA ligase joins those hydrogen bonds to join the fragments together. And DNA polymerase then joins the sugar phosphate backbone to make one whole strand of DNA. So we have DNA helicase to break the hydrogen bonds, DNA ligase to join our two strands together, and DNA polymerase to join the sugar phosphate backbone. Okay, so each DNA strand is used as a template. So I have my DNA over there, and I break my hydrogen bonds with that helicase, and I end up with one strand on that side and one strand on that side. So now this strand is called the template on each side. So now the template gets used and we bring in all of our DNA nucleotides and they all link up, and then we end up with two identical copies the same. So we have one new strand on each original old template. So then this one is going to split again. So is this one. For the next time we're going to have cell division. So when that splits, you're going to have one strand on that side, but now that new strand then becomes the template. And then we make new strands out of those. And the same occurs on this side, where we have that one, and then we make our new strands. So as you can see, each time we use our original strand as the template, and then we break them up, and each time you can see the original strand gets used and used and used, but we keep making more of those new strands. So this is what we call semi-conservative replication. Because the original strand is conserved and used as a template for the new strand. So each new strand has half of the old strand in it that was used as a template. And that's why it is semi-conservative through the whole thing. So that's our process. of DNA replication and our DNA structure.